Good morning. My name is Saurabh Saha. I'm the Senior Vice President and Global Head of Translational Medicine at Bristol-Myers Squibb. Very excited to be in Boston, and even more so that Bristol-Myers Squibb will be opening a research center in Cambridge in Kendall Square later this year. We can't think of a better place to be than at the nexus of healthcare innovation, where we're going to be working very closely with venture capitalists, biotech, pharma, and academia. Now, we're here to talk about precision medicine. And over the last 2 years or 3-4 years, we've seen an incredible surge in precision medicine. We're now able to look at disease on an individual patient level and deconvolute the drivers that are causing the pathogenesis of that disease. Now, what we're able to do is look at those drivers and develop drugs, the right drugs, to give to the right patients at the right time. I'm really happy to be joined by Michael Gladstone, who's a principal at Atlas Venture. Atlas Venture is a venture capital firm based in Cambridge, which has been involved in starting a number of transformative biotech companies over the last 2 decades. We're here to discuss precision medicine and the latest advances we foresee in the future. So, Michael, precision medicine is a broad area. We have genomics, data analytics, bioinformatics, pathology, imaging, just a whole number of technologies that we can deploy to study how patients can best benefit from medicines. What are you looking at Atlas in terms of new innovations in the space?
Sure. So it's a good question. Thank you, Saurabh. Thank you all for having me here. Just a little bit of background on my firm. What we do is we start and invest and back early-stage therapeutics companies trying to develop very novel therapeutic interventions for patients. And so I think as you've alluded to, there's been a massive proliferation and refinement of all the different analytical tools that we have to characterize biology and characterize clinical biology in a clinical setting. And what that's done is it's really shined a light on and a magnifying glass on clinical medicine. It's allowed us to see that diseases we've historically considered sort of monolithic, like lung cancer, are actually not one disease, but a panoply of diverse, heterogeneous subpopulations where any given lung cancer patient may have very different underlying pathological and biological drivers. And so that on its own is not precision medicine. That's just categorization. And that categorization is interesting. It's helpful. It publishes papers. Academics can write about it. But to make that actually useful, and for our companies to make that useful, we really need to understand how that biology maps to selecting or developing an ideal therapeutic intervention for those patients. Otherwise, it's just characterization for the sake of characterization. So what our new companies will often do is they'll focus on a subset or several subsets of those patients and really try to understand at the biological level how that characterization is actually linked to clinical outcomes that we want to improve. And so that usually means going into the lab and using a variety of preclinical models and clinical samples to understand what's really the direction of causality. Why does that molecular trait seem to be associated with poor clinical outcomes? And so what can we do to rectify that directly and have a therapeutic intervention that we can develop in a targeted patient population where we can make sure we are matching the right drug to the right patient, rather than just looking and saying, well, this patient has low T-cell infiltration, but I'm not sure what I can do about that. So you've been on the other side of developing drugs and discovering drugs in the setting of small biotech companies. Now at a much larger platform, really overseeing a deep pipeline that extends across a variety of therapeutic areas at Bristol-Myers. How does precision medicine come into being for you?
Yeah, so at Bristol-Myers, we've had some tremendous success in the immuno-oncology realm, being able to harness a patient's immune system to fight cancer. Now, today what we're seeing is we're evolving away from a one-size-fits-all type of therapy, one that was deployed 30, 40 years ago where most patients would get some sort of chemotherapy regimen for their cancer. Now we're seeing a more precise approach to treating that patient's cancer. So our particular focus at Bristol-Myers Squibb is to incorporate translational medicine across research and development to be able to analyze a patient's cancer and match the appropriate therapy for that patient. Now, when we test a new drug in the phase 1 setting or in clinical trials, we like to analyze the patient's tumor sample, in the case of cancer, and blood sample as thoroughly as we can. And we try to identify markers that may predict whether a patient will respond or maybe a patient won't respond. And then we test those markers prospectively in later stage trials to understand if we can bucket patients into those who will be responders versus those who will not be responders. Now, Michael, we've seen precision medicine take a strong foothold in cancer. Why is that the case, and might we expect that precision medicine will then now permeate into other disease areas across healthcare?
Sure. So I think it's not an accident, it's not a coincidence that oncology is where we've probably seen the greatest development and validation and clinical use for precision medicine. The modern biomarker age was largely rooted 10, 20 years ago in the genomics area. So really at the forefront of characterizing individual patients. That all sort of started at the level of sequencing DNA and being able to associate aberrant DNA sequences or mutations with undesirable clinical situations or a poor response to standard of care therapies. And so it's not an accident that in oncology, where cancer typically is really considered caused by and characterized by a variety of aberrant mutations in the patient's DNA, it's unsurprising to me anyway, that is where we've really been able to see this take root and become clinically useful. And so now, after 10, 20 years of the genomic age playing its way through, we have targeted therapies that intervene on a variety of cancer-associated oncogenes. And so now in clinical practice, it's actually quite feasible, and for a number of patients with a number of different types of tumors, is actually clinically implicated that you should sequence the patient's DNA, and you can actually then have a number of different mutations that are actually clinically therapeutically actionable on the basis of the pharmacopoeia that we've developed, and we can really match certain patients to certain therapies. And so, unfortunately, I think this breakthrough at the DNA level has certainly lagged a little bit in other indications. I think now, though, with the benefit of hindsight and the rapidly decreasing cost of DNA sequencing, we're starting to see similar inroads made in other complex heterogeneous diseases, in my view, particularly affecting the immune system and the neurological system. And so the next frontier we'll be moving into is better defining subgroups of patients with autoimmune disease and neurodegenerative diseases, things like lupus and Alzheimer's, where we can actually stratify patients and better understand in this relatively complex heterogeneous disease, is there a more homogeneous patient population where a targeted therapeutic could be more beneficial than it would be across the broad, unselected population.
Those are really excellent points. And what I find fascinating is if you look at cancer and autoimmune disease, they're essentially two sides of the same coin. In cancer, we're trying to harness the immune system to fight tumor cells. And in autoimmune disease, we're trying to suppress T cells that normally destroy normal body tissue. So what we're learning from cancer, we may be able to apply and fine-tune those pathways that are causing these T cells to destroy normal tissue, so the learnings from one disease area can be applied to another. Now, we have to think about the future. So where is precision medicine going in the next 12 months or even 2 or 3 years from now? And what are some of the biggest challenges and opportunities that the space sees?
So, to take a step back, I think what's worth admiring and being so excited about is that the clinical implementation of these targeted strategies of precision medicine, of actually selecting prospectively a subset of patients and saying, I think they're going to benefit more from this therapy, and I think this other group's going to benefit more from that therapy. We're really in the early days in a lot of settings from clinically testing and validating that. And so I think over the next 6 to 12 months and the decade thereafter, we're going to be able to empirically turn over a lot more cards in the clinic and see where some of those hypothesized targeted interventions will really have the clinical benefit that we believe they will and hope they will. I think the challenge, however, is just that that's still, in many cases, going to take some degree of clinical empiricism, and there's a lot of really interesting hypotheses that need to be tested, and some of this will just take some period of years before we're able to really refine how we treat each of those given patient populations. And so there's going to be a period here where we're going to learn an awful lot, but I think it's going to be a period of iterative refinement in that space.
You mentioned clinical empiricism. Now that resonates because one of the biggest challenges and opportunities that I see, which is a bit of a paradox, is that we've done an incredible job excelling at understanding the targets and pathways that are involved in, let's say, a disease like cancer, and we've done a really good job in research developing molecules that can modulate those pathways. However, we're outpacing, our ability to do that research is outpacing our ability to actually test these with the speed and efficiency and high probability of success in the clinic in patients. I think we need to do a better job, and I think the folks in the room here will actively be participating across various disciplines in coming up with innovative ways to do clinical trials to be able to test the huge combinatorial space of drugs that can help these patients. So with that, Michael, thank you for joining, and I thank everyone in the audience for listening. We look forward to telling you in the next 2 or 3 years how much advances we've made in the space of precision medicine. Thank you very much.